Okay, okay. So I don't know if you listened to the video lecture of going over Hardy Weinberg in the book.
Yeah, I watched it.
Okay, well, it's right there in the book on starting on page 256 and going to 257. When it talks about analyzing gene pools. Um, and so what we can look at, and it's always going to apply to, I suppose it could apply to any locus, but we're only going to use it to look at a Mendelian locus. That is a locus that only has two alleles, and one is completely dominant and one is completely recessive. And so if you take so there's only two alleles out there in the population, and it's as if you took all the alleles from all the members of the population and put them in a big pot. What percent of them would be the big letter, and what percent would be the small letter? And they have to add up to 100% because there's only two possibilities, two alleles. So that's where the P and Q come from. On page 250, 60, frequency of one allele is P, and that's actually the dominant allele. The frequency of the dominant allele is P, plus the frequency of the recessive allele is Q. It says alternatively, but we're always going to say it's P is dominant, Q is recessive. And as it shows on that page, you add those two together, equals 1. Right. So here's the thing. You can, if you know one of these, you can tell what percent of the population is homozygous dominant, heterozygous, and of course, homozygous recessive. And that equation is on page 257, P squared plus 2PQ plus Q squared. So P squared is the percent of the population that is homozygous dominant. 2PQ is the percent of the population that is heterozygous. And Q squared is the percent of the population that's homozygous recessive. Okay. And you can see an example in 1315. If P is 0.8, that means 80% of the alleles in this population are the big letter. And R is therefore has to be 0.2, 20%. Then you do a Punnett square and find out that P squared is 0.64, so 64% of the population is homozygous dominant. 0.2 times 0.2 is 0.04. 4% are homozygous recessive. 0.8 times 0.2 is 0.16. Times 2 is 0.32. 32% of the population is heterozygous. Okay, so and oftentimes you're interested in how many people are heterozygous. It could be a bad allele, and you want to know where how many individuals are carrying this this bad allele. And so so the problem would be find the number of the percent or the number of people in the population or numbers of individuals in the population that are heterozygous. Well, you'll always be given a way in the problem to come up with Q. Right? And as a matter of fact, so you can just watch this video again, or do what I did when I had to relearn Hardy Weinberg. We didn't teach Hardy Weinberg in 106 for a long time, and then we all of a sudden changed the format of the course a little bit, and the other instructors thought we should teach Hardy Weinberg. So I had Hardy Weinberg, but it had been so long, I had forgotten how to do it. So I just went to YouTube. So where am I going to put this? And just typed in Hardy Weinberg. and relearned how to, and he said, you always have a way to find Q. Okay, and this is how you find Q. It'll always tell you what percent of the population or how many individuals exhibit this condition. Because if it's a completely recessive condition, then the individuals that express it have to have two small letters, right? So you know what their genotype is just by looking at them. They're expressing this condition. So you can use that to find Q. So the example on page 257, an actual example of public health officials wanted to know how many people in the population, in the human population, or in, I guess, the US population, that are carriers for PKU, phenylketonuria, which is a recessive condition where you can't break down phenylalanine. And if it's not treated, it ends up with massive mental retardation, and but it's treatable, and we're all tested for it. Within moments of birth, we're tested for it. Anyway, they knew that the frequency of babies, the number of babies born with PKU is 1 for every 10,000 births. So those babies are two small letters. So that's Q squared. So Q squared equals 0.0001, or 1/10,000 of the population. That's Q squared. So how do you find Q? Take the square root. So you pull out a calculator and put in 0.0001, and then hit square root, and you get, whoops, I did it incorrectly. First you got to push the square root, then they go 0.000. Putting out, use my own calculator, equals 0.01. So Q is now 0.01, which is 1%. So 1% of the general population. So 1% of the whole gene pool is a small letter. That means P, 99%. So now we have P and Q. So now we can find out how many people are carriers for it. You can't see from the outside what percent of the population are carriers for it, which is 2PQ. So if you multiply 0.01 times 0.99, and then multiply that by 2, you get, and they have it right there, 0.0198, which rounds really close to 0.02. So it looks like 2% of the population are carriers for PKU. Do you see that?
Yes, and I watched that in the last video lecture.
Yeah, I said the same thing. But let me give you an example. Now that's a real life example, but there'll be a made up one on the exam, and there'll be at least one problem, and there'll be a couple more other questions on Hardy Weinberg. Let me show you one I used to give the class. I used to have group activities in the class, and I used to hand this out and have the whole class, practice of Hardy Weinberg, but we can't do that anymore. So here is the problem that I made up for Hardy Weinberg. And a similar one on the exam. Okay. Down here at the bottom of the page. In meerkats, which are this animal that, like a weasel relative that lives in Africa, there's a locus that controls tail position that has 2 alleles. Big T, which results in animals with an erect tail, and small t, which codes for protein that causes a drooping tail. So the small t is recessive. The erect tail is dominant, or complete dominance. In one meerkat colony scientists counted 812 animals. And they noticed that 130 of them had a drooping tail. Calculate for them how many animals in this population are carriers for drooping tail. Do you think you could do that?
I think so.
Give it a shot. I'll help you out. So you got to find Q first.
So would Q be 0.4? So, um,
Uh,
percent.
well, not 0.4%, but hang on a second. Okay, so you came close. So what's the first thing you did?
So I did 130 divided by 812.
Correct. And you got 0.16, right?
Yes.
Okay. What's the square root of 0.16?
Is it 0.4?
Oh, yeah, you're right. I thought you said 0.04. You're correct. You got it right. 0.4. I don't know. I heard it incorrectly. Okay. So then so that's Q. That's Q. So what is P?
So wouldn't that be 60% because it's the other half?
Right, so 0.6.
Yes.
Okay. So now, what percent of the population are heterozygous?
Okay, so, and that's the 2 times PQ, right?
2PQ, right.
So 48%?
Right. So there's one part left to the problem. What's the problem actually looking for?
How many animals in this population are carriers for drooping tail?
Right. So how many animals are in this population of carriers?
Okay. So would it be, oh, I did that wrong. So would it be around 389?
Yeah, well, round it to 390 because you can't have 3/4 of an animal. So you round to about, so 390, that's correct. That's how you do it.
Okay.
All right, the other important thing, so that's how to do the problem. The other important thing about Hardy Weinberg is, it has to do, and this has to do with microevolution. I think I mentioned in the video what microevolution is changes in allele frequency from generation to generation. So like here it's Q is 0.4 and P is 0.6. Well, what if something happened to the to the meerkats with the drooping tail somehow died more often through either natural selection or but just by genetic drift, and more of them died than the other ones carrying a big letter. So in the next generation, the percent of the percent of Q, the small letter, went down to 0.3. That means the percent of the big letter went up to 0.7. That is a change in allele frequency from one generation to the next. For either adaptive reasons, natural selection, or because there's something happened randomly to the ones with the drooping tail, they died more readily or something, so it changed. That is microevolution. And then if that goes on for hundreds, thousands, hundreds of thousands of generations, then eventually, through a microevolution, two groups of the same population could spread apart and end up becoming reproductively isolated and therefore becoming two different species. And that's called macroevolution. But first you have to have microevolution before you can have macroevolution. Of course, some people don't agree with the creationists, say, okay, they agree with microevolution. I mean, it's observable. You can see allele frequencies change from generation to generation. But becoming whole species, different species, is such a slow process, you can't see it. So you have to infer it, and some people don't buy it. Anyway, but here's the thing. What if at this locus, this drooping tail locus, it didn't change? The allele frequency didn't change. The next generation, Q is 0.4, P is 0.6 still. That would mean that's called Hardy Weinberg equilibrium. That means the allele frequencies are not changing and there's no evolution going on at that locus, because evolution is a change, from generation to generation. So if it doesn't change, so if this allele frequencies in this locus doesn't change, it's in Hardy Weinberg equilibrium, and there's no evolution going on at that locus. So does that make sense?
Yes.
Yeah. And I think that was it on Hardy Weinberg. Sounds like you got Hardy Weinberg down.
Yeah, I think I'm good on that. And is that the only question that will be on the exam like that?
Well, there could be questions on, I'll give you a scenario on, it's a good question, the different types of natural selection. On page 264, directional selection, disruptive selection, and stabilizing selection.
Oh, and that's the one with the graphs and the peaks, right?
Right. So you have a locus that's controlled by more than one gene. So now we're going non-Mendelian, and we have a normal distribution. And so they were having the coat color in mice. It goes from really light gray to medium gray to really dark, almost black. So that's the distribution of phenotypes. From the average color is like a medium gray. Right? Directional selection moves the bell-shaped curve to the left or the right. Doesn't change the shape of the curve, it just moves it to the left or right. Somehow a slightly lighter color becomes more favored and they survive more often than the ones with the darker color, or the darker color becomes more favored, and they become selected for. So generation, the bell-shaped curve moves toward the dark end. A good example, old example of this is called industrial melanism. In England, they had all these trees with light-colored bark, and as this moth lived on this tree, or often went on this tree, and they were light gray colored, but there was there's a gradation of coloration in every organism. In every phenotype, there's every characteristic, there's a range. So they also had very dark colored moths and very light colored moths. Well, this was directional selection toward the light color because the light color blended in with the bark. It was hard for birds to see, but the dark colored moths just stood right out and got picked up all the time. Then the Industrial Revolution happened and all this soot came pouring out of these smokestacks, turned the trees almost black. So now the darker moths were blending in with the bark and the lighter moths stood out, and that just shifted the whole bell-shaped curve to the dark. So that's directional selection. So that's the favoring of one extreme over the other, favoring of one extreme over the other extreme or the mean. All right. Disruptive selection is a favoring of both extremes at the same time, and not the ones in the middle. So then you get a concave curve. And this was, I think I'll give the example on the video lecture of birds on an island that there's big seeds and there's small seeds. And if you got a big beak, it's not worth it eating the small seeds. And if you got small beak, you can't eat the big seeds. So birds are just eating big seeds, birds eating small seeds, and the ones in the middle with the medium-sized beak are at a disadvantage. And this kind of disruptive selection is the one that can lead to speciation, because you have the two groups moving apart and doing different things and not interacting that much anymore. And they could become different species. So that's important to know. Which selection could lead to speciation, macroevolution? It's disruptive. Then stabilizing selection is the mean is favored over the two extremes. And what this does is just change the shape of the bell-shaped curve. It becomes narrower and higher and narrower. And a good example of this is human baby weights. Really low weight babies who are born with really low weight are more likely to die than babies that have the mean weight of human babies, I think it's like 7.5 or close to 8 ounces, pounds. And then really heavy babies that are born also have a higher death rate, the high mortality rate. So the healthiest babies are the ones that are right in the middle of the range, and that's why they get taller and that's called stabilizing selection. Oh, and I remember also, the mechanisms of evolution, we talked about natural selection, and we actually do that in lab. But there's genetic drift also, the bottleneck effect and the founder effect. And there's 3 types of selection. Natural selection, that Darwin talked about. Artificial selection, which is what we have done to modify the uh, plants and animals we live with and the plants and animals that we eat. And then there's sexual selection. That's on page 263, which goes along with sexual dimorphism, which is a species of organism that you can tell the two genders look different. Just by looking at them, you can tell which individual is male, which individual is female. So obviously human beings are sexually dimorphic species. We have different genitals. So even a really young person can tell the gender, but after puberty, we get secondary sexual characteristics. And then that changes the appearance. Now, and actually the more extreme the sexual selection, so it's more extreme the sexual dimorphism, often the more extreme the sexual selection. And that is the females select what males they are going to breed with. And these males have to have certain characteristics or else they won't breed with them. Now there's some organisms that have no sexual dimorphism. For example, robins. You can't tell a male robin from a female robin just by looking at them. But you can't even tell if you capture one and you spread the feathers apart and try to, they don't have external genitals. They have a cloaca and with 3 openings in it. And male cloacas and female cloacas look exactly the same, so you can't tell. So there's no sexual selection in those kind of species that don't have sexual dimorphism. So that's a effect, and it's really interesting. I mentioned, I don't, if you listen to the video, I mentioned that scientists often wonder why females are selecting these things in males that doesn't seem to be make any difference, or actually makes things harder for the male. So it's actually like give the male a deficit and see whether he can survive, find enough food, be healthy, survive long enough. And if that's the case, maybe he has good genes. If I mate with him and I make male babies, they will be successful and they'll pass on my genes. You know, so that's the thought. Now, I got one more. Might be just one more video lecture, one more chapter about how biological diversity evolves. That's chapter 14. We're going to do that, make it that this week. And it's not going to be the whole chapter, but it really talks about how, well, it might be actually close to the whole chapter, how evolution really works. You know, what is a species? How does macroevolution? So the chapter 13 deals a lot with microevolution, talks some, but macroevolution is really chapter 14. How does one species become more than one species? And then how do species remain species by not making the ultimate mistake, and that is trying to mate with somebody who's not the same species? That's usually not going to work. And there are all kinds of ways that organisms make sure that they don't make that mistake. So I'm going to go over that. Chapter 14 this week, and that's going to be the end of the video lectures and everything for the exam. So the final exam is just going to be chapters 13 and 14.
And will the exam be released the beginning of next week?
Yeah. I think finals week starts on Tuesday, I think, doesn't it? And it goes to the following Monday. You see that? Use the calendar. Today's the 19th. I think 26th in 1 week is the last regular day of the semester, and then 27th through the 3rd of May, I believe, is finals week.
Okay.
So I'll have it open all that week.
I don't think I have any more questions.
Okay.
Thank you for answering some of mine.
Yeah, that's what these. That's what I wanted to have these Zoom meetings for. You know.
Yeah,
Q&A.
the last one was really helpful before the exam.
Oh, the one dealing with the non-Mendelian genetics problems.
Yes,
Yeah,
that
yeah,
helped a lot.
yeah, yeah.
All right. Bye.
Okay, so long. Have a good one. Bye. Well, that's going to be it. So let's stop recording.